we're going to start off with a great talk from Robert Thacker. He is your Amnis guy, okay? He does all things imaging stream. He's been with Amnis longer than Amnis has been around. They've been bought they've been bought by lots of companies, but he is your Amnis guy. He knows everything about the image stream. And he is a guru on that. Obviously, he did do a PhD and a postdoc. Those were in Cincinnati, and during all that time, he also, he did a lot of cell-associated antigens with dendritic cell subsets, so that's pretty cool. We like dendritic cells here. And he became an application scientist with Amnis back in 2012. So like I said, he is your Amnis guy. He's going to talk to us about a technology called cell painting. So let's give a round of applause for Rob.
Good morning. I've been told my voice is kind of nice to fall asleep to, so hopefully that's not what happens this morning. So, and keep you engaged. Thank you for the introduction. Thank you for the opportunity to be here. I've been doing image stream. I used the image stream during my postdoc at Cincinnati Children's, so been around it a long time. Disclaimers, and yes, I am employed by Cytec, who is the manufacturer of the image stream. So, in case you didn't know that, but here we are. So, in preparing this talk, it's like a lot of people know what the image stream is, and a lot of people still amazingly don't. We've been around for about 20 years, and the foundational technology is still kind of that same thing. So I think it's important to kind of talk initially about imaging flow. Obviously, we have a lot of flow that's been happening in this conference, some imaging flow that's been happening in this conference. But as scientists, you are wanting to understand what's going on within cells usually, right? Sometimes you want to know the tiny things, maybe EVs, exosomes, all that stuff. But in the end, we're trying to understand how our biology is working, how cells are interacting with each other, how things are interacting with those cells, maybe where things are within those cells, expression. And so for flow, we're usually using that because we want to know molecule expression, population statistics. How is that fluorescence, that marker, changing and shifting within these populations? If you need context, though, where is that signal coming from? Historically, we've turned to microscopy, right? Because you can get different information there. Size, shape, location. Ask questions like colocalization. Are these two markers in the same space? And that's where the image stream and imaging flow cytometry sits. It's kind of this bridge, a third leg of the stool, if you will, in this space, because it allows you to do higher population statistics, but you have location information, you have context. And as an example, here we have a panel that's 1, 2, 3, 4, 5 different colors, 5 different subsets. And we have a green population that pops out and a red population that pops out. And then the rest are kind of just sitting down here below. But the context that's provided is that by adding in some different markers, in this case for a lysosome and your molecule of interest that you're wondering where it's trafficking, you now can see that within a single tube, the green population is behaving differently. There's localization that's happening within the lysosome that is not happening within the red population. And that's really one of the true strengths of adding flow and imaging together, is you're now able to do this immunophenotyping and understand that cells behave differently even if they're sitting within the same space. And they're doing different things, and you can do different functional assays to understand that. And so I've seen time and time again, even though we have, I've been with the company for a while through multiple different exchanges, but it really is, I see time and time again how it enables applications that are challenging to do just by flow or by microscopy alone, right? It's not the end all be all, but it is definitely a tool that I have found to be extremely valuable and to help people answer questions that sometimes they've struggled to answer for years. And it just really is that tool that can do that. So how it works. The foundation is that it is a CCD detector, and there are multiple channels within that CCD detector, so it's essentially spread out. You have an actual microscope objective lens, bright field LEDs to get your bright field images, and then your bank of lasers. So that emitted light goes through a 20, 40, or 60x objective lens, and then goes down and gets split apart so that you're simultaneously collecting all of those different channels and your bright field image at the same time. And this is kind of what it looks like. This is what side scatter looks like when you take a picture of it. Pretty regularly, people run samples and they're like, what is that? I'm like, that's your side scatter, right? That's what it looks like when you have an image. Bright field images and then your different fluorescent channels. This is what a surface stain looks like. This is what a nuclear stain looks like. And this is NF kappa B in this case, where it's kind of cytoplasmic in this particular example. Right, but that is the thing, is it preserves that spatial location so that you know where it's coming from. Also foundational to this is time-delayed integration. Because it is a CCD device, we're able to use this time-delayed integration. And what you notice is, here's you have your lasers, your laser, it's interrogating the cell or whatever is going through there, and you can see that it's getting brighter and brighter as it goes down. It's literally tracking it digitally so that that light is being transferred down pixel by pixel row, and then it's integrated into a singular event and image at the bottom. Makes it so it's not streaky. It increases our photon collection time so that it's more sensitive. And all those objects, it's synchronized, right, across all of those different channels. And so it does combine the high throughput. Some of you are like, I've used it. It's not so fast as a flow cytometer, right? It is a slow flow cytometer, but it's a really fast imaging system, right? Like there are very, very few, if any, other systems out there that image as quickly as the image stream. Because if you're collecting, say, even just at 1,000 cells per second, that'd be 12,000 images per second on a 12,000 or on a 12-channel system across all of those different channels. And at higher speeds, you can go up to 5,000 objects per second. It is 20, 40, 60x. We have things like extended depth of field, various lasers. And I think one of the real benefits is it really digs deep into the imaging part of it with the ability, which can be confusing for many people because it's just like overwhelming with how many options. But some of the tools I'm going to show you today with the machine learning kind of show you how you can tap into those different features without necessarily knowing which one you need to use. But 86 different features, 22 different functional masks that you can change, machine learning and AI, and it really all works together. And I think one of the really nice things is every single dot is connected to an image. So you can click on that dot and you can see the image. You can click on the image and you can see where it is. So also, very important to how the image stream works is you can see this fluorescent image here, you know, your bright field and your different channels. You can see this kind of grid, right? So that's your pixel array that's in the back there in that CCD camera. And the reason this is important is because it means that every single pixel that is collecting light, we have a location associated with it. There's an X and Y coordinates. We know where that is, and it's connected across each and every one of the channels. And that makes it so that if this is a 60x image, this pixel represents 300 nanometers. And within that 300 nanometer space, we can tell if that is bright in green, or if it's bright in red, or if it's bright in blue, or whatever color you have in there. And allows it then to be synchronized across all those different channels so that you can apply compensation also across all of that. And this is what allows you then to do some of these assays to tell differences in textures, differences in location, and then do things like similarity. So if we were to take this image, although you don't ever get this information, this isn't something that you can like pull out of the ideas software. But if you were to invert it and make it on the channel 11, which is a nuclear stain, you would know that it would inverse, right? The inside of that would be high and the outside would be low. And this is what allows us then to tell all those differences, right? As I mentioned, there is a direct link between the images and between the images and the dots. So in this case, we have very first measurement that we often make is an area of the bright field. So sometimes people ask, like, how do you know if you have singlets or doublets? You just look, right? You draw a gate even during acquisition and definitely during analysis, but even during acquisition, and you can draw a gate and you say, what are these guys? And these ones that are smaller in area and circular, which is aspect ratio as a measurement of circularity, are these small singlets. And the large singlets are a little bigger, but still round, and your conjugates are bigger and not so round. And then you can apply it. And then down here is like your debris. So if anybody's ever like, why didn't you collect this stuff? You can draw a gate around it and show them, did you want the garbage? The EV people are like, yes, please, right? It's not garbage. It does lots of stuff. But, and then of course, as I mentioned before, you can connect it with immunophenotyping. So here we have double positive events, which are correlated to the conjugated events and so on and so forth. So, all right. So to get into the topic for the day, I just wanted to, again, give a little bit of an introduction. But this cell painting assay that the team in Seattle worked on, thought it was pretty cool. And I think it also really leverages the benefit of higher resolution imaging flow cytometry and the tools that machine learning provide to be able to allow you to kind of dig into this and notice differences. So cell painting, traditionally it's a high content, multiplexed image-based assay for cytological profiling. It's usually done on a slide. In this case, I want to know, could we adapt it to cells in flow? Could we do it on the image stream? And what happens is you paint or you label different portions of the cell. So the RNA, the DNA, the ER, mitochondria, Golgi, and cytoskeleton. And then the dyes paint those cells and allows you then to look at it and say, what is changing? What is different? Importantly, it does use a standard staining protocol, so you could standardize this across multiple cells. And then you can characterize those cell profiles. How are they before treatment? And did those different compartments change after treatment? And of course, it is going to identify, well, maybe the mitochondrial changed, that image changed, but the nuclear image stayed the same. And so it allows you to kind of see how things would go. And these are 60x images on the image stream. So again, as I said, they're simultaneously collected across the board. So we have a bright field image, and then you have your various markers. And this is the channel that they went in. The idea behind this was if somebody wanted to repeat it, we're more than happy to share kind of how we did it, which markers we used. And so we have Cyto14, which is a nucleoli RNA stain that sits in channel 2. That's like a FIT-C, right? MitoTracker orange, that's in channel 3. That's like where a PE would sit. Con-A, LX4 594, AF594, the ER, channel 4, right? So same as like a PE Texas Red. And then CD45 is a PE Sci5. That's in like channel 5. So this is all what would sit on like the first camera of the image stream, right? Those first 6 channels. And then when you move to the second camera, this is where our 405 and 642 lasers sit. You have your DNA stain in channel 7, your WGA, which is Golgi in channel 11, which would be far red, like an Alexa 647. Oh, that's where it's sitting right there. Here we go. And then the further red, Alexa 750. That's all the way out in what we would say is channel 12, right? So this is all simultaneously collected. And then we did it at 40x and 60x. These images are 60x. This is just showing you the laser powers and things like that to show you if somebody wanted to repeat it, how we did it, right? And then we do have side scatter that's in there as well. So the way that these cells were treated is we have fenformin, which is a biguanide. It was used in type 2 diabetes treatment, but they removed it from the pharma market because of it induced high lactic acidosis and inhibition of the mitochondrial complex. And so we took this, had different doses, cells were treated for 72 hours, and then they were analyzed. They did the cell painting staining, and then they were analyzed with machine learning. And then this is like the foundation, right? We have the IDEAS software, and then for those of you that ever use the IDEAS software, this is kind of that foundational. You can create masks, you can create features, but then there are also wizards and other tools in there. But the machine learning is something that in the last several years has really, I think, expanded the ability or what the image stream can do, because it taps into your ability to say, this population looks like this, and this one looks like this, and this one looks like this. And you can ask anybody in the lab to say, could you just make an image gallery of these, of these, of these, right? And essentially, when you just do this without the machine learning, you do notice differences, right? So here we have a mitochondrial stain. If everybody looks at 0 at the treatment of 0, you can see that that's kind of just a smear. It's kind of equally distributed throughout. At 2 millimolar, you can see these puncta that are showing up. So the mitochondria are doing something different, right? It's staining differently. And we can use a singular feature, bright detail intensity in this case. And you can see that there is this shift, right? Bright detail intensity is just saying, are there brighter pixels within this image? And there are, right? We have brighter pixels that are there. Over here, if we look at the nuclear stain, we can see that it definitely changes. It's probably measuring apoptosis, that those cells are beginning to fragment and break apart. And that by itself can also be measured, right? We can say, all right, let's use that as down the line, we see that the apoptosis increases, right? Increased texture, aggregate formation, and granular formation. And so why would we then use machine learning? We already have two features that are kind of like showing some differences here, right? But if we just had one feature here and another feature here, where would you draw that line? And it can be challenging. If I didn't have the yellow and the blue in here, you'd be like, I don't know, where do I draw that line? And if you use a singular feature, there's still kind of this crossover that can happen, right? And so what happens is with the machine learning, it combines features. It makes it so that this classifier isn't just a singular feature, but it can be made up of 2, 3, 10, 700 different imaging features that are then all equalized and put on a single plane, right? And as an example here, we have actin polarization, right? And so this was a truth set, this was the truth set, and this is the classifier, and 0 becomes the pivot point, okay? And I explain a little bit more about kind of how this works. So you have the input of the truth sets, you go through this process, and the features are normalized to a common base to offset the wide variation in feature value ranges, because some are, on a scale, they're just on different scales. So it normalizes them and then uses Fisher's discriminant ratio to indicate which population or which features are the best, right? And then it ranks them. But you see you have negative numbers and positive numbers. In the end, the classifier doesn't care. If it's positive or negative, it normalizes them all and kind of combines them together, right? And so you can see that if 0 becomes the pivot point, this was normal, and then if things shifted, we have a positive score. If you flipped it, obviously they would just go the other way. Here's the math behind it, but in the take-home message from this is simply that a higher RD value is better, right? If you have a singular feature with just 3.9 or 3.4, and the machine classifier is 4.28, you're going to get better separation. And even just these little tiny ones, like 1.29, 1.23, 1.39, can make a difference, right? That little bit of shift. We talk about resolution all the time and seeing things from background, and sometimes just that tiniest little bit of shift can make a population more clear. So how does this apply then to the cell painting? Well, what happened is we took those treated and untreated, highest dose, lowest dose, fed it into the machine learning, and said, look at these different signals. Look at the mitochondria signal. Look at the nuclear signal. Look at the ER signal. It created and screened 2,810 different features. I didn't have to create them myself. The machine learning software did it. Of that, it said 62 of those features are actually giving us a meaningful difference. And it then takes this, and if you look, it's color-coded according to which portion of the cell it affected, right? And so the purple is the DNA region, so a lot of the nuclear changes happened here. The green is the RNA, the yellow is the mitochondrial changes, and you can see then these shifts that are happening within those cells. Now you take all 62 of these features and combine them into a single classifier that I can now plot on one line, right? And this is what you get, right? Is untreated is below 0, treated is high, above 0, between 1 and 2. And you can see the corresponding shifts in the mitochondria, in the DNA, in the nuclear images, and all of this, right? And so it really allows you then to now do this and even do a dose response. And you can see that at 0, it's below 0, it's below that 0 line. And as you increase the dose, that line shifts over because you have more and more changes that are measured. And I just think it's a really powerful ability. Someone didn't have to go in and gate hundreds of different gates and masks and features. I've seen some pretty crazy analysis out there, but this could have been done by, I mean, maybe not the staining by a grad student, because it could be a challenging staining protocol, but this could be done by somebody who said, all right, I did this, I fed it in machine learning software, and I have my shift and my change. So it really unlocks the power of the imaging. And this isn't just cell painting. We do it all the time for simple things like doublets, singlets, clumps of cells, changes in nuclear morphology, changes in cell morphology. And so it's really, really powerful. So The last thing I want to say is that it is because of the higher resolution imaging that this is able to be done, because that 40 or 60x really gives us that pixel by pixel definition in order to be able to measure and see these shifts and changes. So with that, really want to thank the team in Seattle who did this. I'm just presenting it today. I didn't have anything to do with the other part of it, but thank you so much for having me here. And if you have any questions, please come talk to me. Or you can ask me now. Yes. Yes, I can. Yeah.
Can it comes in combination with IDEA 6.2 version, or I have to use IDEA
You have to use 6.4.
6.4?
Yes. Yeah.
6.2?
I can give it, yeah, 6.3 it'll work, but the only difference between 6.3 and 6.4 is Cytec branding. So let's just get you 6.4. So yeah.
I believe that I can use as many as 10 colors with machine learning.
Yeah, you could use all the channels. Absolutely. Yep. And we have used it actually to pull out some of those immune synapses as well, because sometimes that's challenging just to find them.
Side scatter parameter for a different color. And
yeah.
But if you can elaborate a little bit more, the use of side scatter as side scatter.
Yes.
I think it would be very helpful to.
Yeah, so in that case, it is one of the parameters you can choose, right? And it won't just look at intensity of side scatter, but it will also look at texture and pattern and all of those things. So I have used side scatter much more regularly now that I use machine learning on a regular basis. Historically, I was like, yeah, area aspect ratio, got my singles, doublets, we're good. But side scatter has become much more useful now that I have the machine learning. Absolutely. So, yeah.
Thanks, Rob.
All right.